Yeah, hi, I'm Bob Deans. I'm CSO at Synthego, and I'm real excited to be here today. I'm excited to tell our story to you, but I'm also excited because I really love Synthego. It's been an incredibly uh, innovative and compelling company to join up with. So at Synthego, our ambition is to make accessible genetic medicines routine, and we're about that by engineering biology. And that's going to be a recurrent theme as I present our data sets and our goals to you. And so let me go ahead and give you a background on Synthego, and you'll understand better when I describe engineering biology, what really the DNA and the roots of the company are. And so Synthego was founded by two brothers, former SpaceX engineers, Paul and Michael Dabrowski, and has grown to about 250 scientists altogether, after launching in 2016. One of the real strengths of the company has been the scientific advisory board that we've built, the advisory board around Paul and Michael. And you can see here, going clockwise from Jennifer Doudna, Matt Porteous, Bill Scarnes, Sir Andrew Whitty have all been really key advisors to the company. And so the company launched as a genome engineering company in the beginning, building standardized CRISPR reagents, but quickly moving to producing rapid, large-scale experimental workflows that really speed up discovery and time to clinic. And so they've done that in a very transparent way, and I think it's really laudatory to understand how Synthego and Synthego scientists have reached out into the community and really supported transparent innovation and discovery efforts. So at Synthego, we've built what we call a full stack biological insight factory, and I'm going to talk to you a little bit more about it. And so what you see in the middle of the slide here are really the technology stacks that we've assembled that allow us to go from early discovery and accelerate into the clinic. That's based around building capabilities in RNA synthesis that include GMP capacity. It involves engineering cells to serve as screening models. It involves computational biology and screening tools we use to be able to interrogate rare disease alleles and functional biology. And it caps off with a bioinformatics and computational biology platform that really lets us model disease and integrate the discovery platforms in a really productive way. So really what we're doing when I talk about turning biology into engineering is automating each of these stacks that I've described to you, turning each stack in the value chain into an automated high throughput process. And so we believe that this moving in biologics to engineering is highly transformative and disruptive. And what I'm illustrating here is really how dramatically the molecular medicines, genetic medicines, can flip the standard discovery pipeline that you see on the left for drugs or biologics upside down. And it does that for a number of reasons. One of those reasons is the accuracy that's implicit in employing artificial intelligence, machine learning, and gene modeling to improve target discovery, and additionally stratify patients so that your target is suited for a patient with high likelihood of response or in the right demographic and background to receive therapy. It's also very powerful because that same link between gene and disease also gives you a powerful tool set of analytics that let you describe potency and interrogate that clinically. So we're improving accuracy in target discovery, we're improving accuracy in clinical readout. On top of that, we're all aware that accelerated regulatory approval paths now exist. And many of those allow approval based on interrogation of gene-based surrogate omics or gene analytics. So what you're seeing is a very great pipeline to move directly from target ID out to real-world patient clinical access data. So, I'm building the story around genome engineering and engineering biology, and I just want to reinforce that the core of our business is genetic diversity. It's reading it, writing it, and developing new classes of these medicines. And so I just want to pose what's limiting us in translating some of these genome engineering tools into the clinic. Some of that is an uncharted regulatory path that we're not fully conversant with the risk and the regulated evaluation of the products that are coming forward in this new territory. So another angle is definitely around safety and precision of the tools that we're using in terms of introducing cytogenetic risk or evaluating cytogenetic risk. And implicit with that is the need to see standards arise in this space involving the analytics we use and the algorithms we use for analysis. So I want to walk you through the development cascade that Synthego has built. And I'm going to begin back in 2016 with first products coming out. Really the core platform, this is our Halo platform, is the ability to make the synthetic guide RNAs. And so Synthego elevated that capacity to include the ability to make multi-guide library screens. So the ability to make high efficient knockout libraries by employing guides in a target cell is really the first stage of the discovery process. So building upon that, Synthego moved quickly to be able to produce screen-ready cells, engineered cells modified by CRISPR that replicated and allowed testing in a disease model. So you can see a progressive build from platform to discover to model. And now, as I mentioned before, the enablement by providing GMP guide RNA that sews this together with a rapid, single line of sight entry into the clinic. The real excitement is in the future, and it's really around the computational biology and machine learning tools that we're employing both across this platform, but also in our own thoughts for target discovery. What I'd like to do is move now to some really interesting data in 3 or 4 slides that represent the power of the automated platforms I've just described. And this involves a collaboration that we've undertaken with Nevin Krogan at UCSF. And it's a very early work that Nevin did in response to the COVID pandemic. And so what Nevin and his lab had done is define the interactive proteins in the human genome which were necessary to support virus replication. And they did that by a series of immunoaffinity mass spectroscopy studies. And this was all data that is out and has been published. And what I'm showing you on the right is the interactome map, the viral proteins and the human targets that they interact with. So we were able to connect with Nevin and undertook a model to rapidly screen those human interactome targets for relevance in supporting viral infectivity. And the thinking is that if we could identify those interactome points that were critical for infectivity, we could then apply a drug-based discovery approach to try to interfere with virus replication. And so in many ways, what I'm describing to you in the data set that I'll walk through is really a target validation by gene knockout that is highly impactful on the discovery process. So what we had done, Nevin had identified around 330 different critical genes in the human interactome, and we built panels of guides against each one of those and built independent gene knockout pools representing those knockouts in 3 different cell line backgrounds. And so the majority of these genes could be knocked out stably and represented. Some of those were clearly essential genes and fell out. But the point I want to make here is that in 2 weeks, we synthesized 1,000 different guides and in 2 different cell lines, created over 300 different gene knockout libraries. So it goes much better. The story is really great. So we were able to then take those individual knockout pools and distribute them to a variety of investigators in the COVID space. And the purpose here was to have orthogonal testing on viral infectivity and to understand by spreading the assays out across labs where we had clean reproducibility. So it took us another week after creating the knockout pools to prepare screen-ready cells, ship, and have our first infectivity data come back to us. So I'm describing a 3-week period to get the data that you currently see on the screen, which is really remarkable. And just as a prelude to an upcoming preprint, several of those have gendered reproducible hits in genes critical for infectivity, dropping viral titer by several logs, forgive me. So this is really an understatement, I trust, in having built the story to this stage, that genome engineering can really be a tremendous translational accelerator. I want to take 2 slides and show you how the efficiency underlying gene editing is really an important element of that. And what I'm showing you here is how we can engineer in a very high safety margin, the difference between a desired on-target hit and an undesired off-target hit when performing gene editing. And so the way we approach that is to integrate machine learning and automated processes, a common theme in this story. And what we can do is shown over on the right. This is looking at a standard target for genome editing, which after several condition tries was resting at about 7%. This is sort of a standard university research lab. We'll use their best protocol and get efficiencies in this range. What you can see is that in a period of 3 days, we incorporated 200 different ratios of enzyme and guide and editing conditions, we're able to raise that up to an 82% threshold. So bringing efficiencies up to something that can be impactful when you start to talk about translating into the clinic. In our off-target analysis, you can see below that we get very consistent hits. So long story short, we can automate the process, we can get maximal on-target hits, and we can statistically validate and know repeatedly what we hit on off-target. So this is the way we can make a very compelling target regulatory package for review in order to move translationally into the clinic. I'm going to talk now about some discovery efforts from our chemistry group that have also been very, very exciting for us. And so this is along the theme of improving that on-off target ratio for maximal safety margin when approaching clinical opportunity. And what we're doing here is chemically modifying our guides, and we're modifying them chemically so that they can be blue light inactivated. And so this allows us to control the on-off time for editing in a very prescribed way. That's what you're seeing in the upper left panels here. The difference between editing with and without light, you can see we can essentially look at this in an almost binary on-off manner. And then down below, you're seeing the same data evaluated over time. And the message that I'm delivering here is that we have the ability to prospectively organize an optimal on-off target ratio and really move ourselves directly forward in the translational path. So what I'd like to do now is shift over and talk a little bit about disease modeling employing genome engineering, and so moving us up the stack. And really make the obvious point that in this space, clinical data, particularly at the single-cell RNA-seq level, and the ability to derive patient-specific iPSC has provided excellent training sets for systems biology and understanding disease. So CRISPR editing can assemble known disease variants, rare disease variants, into an iPSC background and further interrogate. And so what you're describing is an example where we can collect patient disease iPSC or generate healthy iPSC and introduce and interrogate the same gene in both backgrounds. So we start to assemble very important information about links of a rare disease variant to other components in gene networks that contribute to pathology or represents targets for intervention. So what I'd like to do is run you in the next couple of slides through a quick example of that. And what I'm showing you is a gene associated with an inborn error of metabolism that has multiple allelic variants, all contributing to different levels of disease, and how we've used that as a model for study. So what's represented in the upper left is a gene map and the positioning of the rare disease alleles across exons. And on the right, you're seeing our strategy in breaking these down individual editing units with the ability to reincorporate each and any of these rare disease alleles into an iPSC background or another target desired type. What you're seeing on the bottom two figures are our ability to generate pools of cells representing those variants, and then also take and singulate or take those pools down into single cell analysis and do further study. And so that's what I'm representing here on this slide is looking at the pools and looking at the single cell variants. And long story short, I'm telling you that the metabolic activity predicted from the patient-driven population is replicated in our rare disease variants. And additionally, if you look to the right, when we further interrogate those into single cell pools, it's beginning to give us the data that we're very intrigued with about the other compensatory changes in the cells that accommodate the rare disease mutation. I think it's a very powerful drug discovery process. All of these data link very logically to what's understood about disease. This is representing the rare disease alleles on the left, and then understanding the biology by having a structure function model that we've been able to plug into. So a very highly informative description of how we can employ screening technologies around rare disease elements. And this is a summation of what I've just described to you as a workflow moving from right to left. It's really about identifying the variants, designing the guides to evaluate them, moving those into a cell background, and then using that cell background, stressing the value of iPSC to really look at functionality in terms of developmental biology or final cell biology. And then the real exciting part is that the interrogation of the gene models, and particularly using current omics technologies and single-cell RNA-seq to move forward. So I believe I've led us through the full stack here, the growth of technology within the company, and then the alignment of that growth around accelerating translation into the clinic. So we think we represent a very powerful partner in the translational process, both on the discovery side, on the manufacturing side, given our ability to manufacture a GMP guide RNA, and just really look forward to more opportunities in this space. So one of the most exciting things was an announcement we made several weeks ago about a recent financial close. And it really gives us the capital to flex our muscles around the platform and show some significant growth. And it also, going back to the original story of tech meets life sciences, it also takes us back in the position of incorporating more life science innovators into the capital pool, with Wellington and RA Capital coming on well to join the VC. So with that, I thank you for the time and messaging today. That's really, the R-MESA meeting is one of my favorite meetings ever, and it's disappointing in one degree to have to do this virtually, but my excitement about growth in the industry and where the market sector is just tells me how much more fun we'll have next year. So thank you all.